When a crime scene looks completely spotless, it rarely is. Even if a suspect spends hours scrubbing a floor with bleach, microscopic traces of biology almost always get left behind. Investigators find these hidden traces by spraying chemicals like luminol or fluorescein over the area and illuminating it with an alternate light source. If there are invisible traces of blood, the area will suddenly emit a ghostly blue glow. This glow is the result of an oxidation-reduction reaction. Blood contains hemoglobin, and within that is a heme molecule holding an iron atom. When the chemical mixture hits the floor, that heme acts as a catalyst. It steals an oxygen atom from the hydrogen peroxide in the spray and hands it over to the luminol, triggering the blue chemiluminescence. But that oxygen theft is also the method's biggest weakness. Common household cleaners, especially bleach, are highly reactive oxidants. They can hijack this exact same chemical reaction and cause the floor to light up, even if no blood is present at all. To run a more targeted check on a specific stain, investigators use colorimetric field tests, like the Kastle-Meyer or benzidine assays. These swab tests rely on the exact same oxygen-stealing mechanism driven by the heme molecule, but instead of emitting new light, the chemical reaction alters the physical structure of a dye on the swab. This changes how the material absorbs and reflects room light, usually turning it a bright pink or a dark blue. Whether they produce light or color, these tests only provide what is called a presumptive positive. They highlight where the crucial evidence is hiding, but they do not explicitly prove that the substance is blood. To physically prove a sample is blood for the courts, forensic labs rely on a century-old technique called the Takayama crystal assay. By treating the suspected blood sample with a mixture of glucose and pyridine, the chemistry forces the iron inside the heme to form new chemical bonds. This physically alters the molecular geometry, building out a rigid, bright pink crystalline structure. Once the substance is confirmed as blood, the lab must prove it came from a human rather than an animal. They do this by throwing the sample into an immunological battlefield using a technique called crossed-over electrophoresis. Applying an electrical current pushes the negatively charged Y-shaped antibodies toward the cathode, while driving the positively charged human antigens straight toward the anode. They are on a direct collision course. If the blood is human, the corresponding molecules will lock together mid-gel. When the lab stains the gel with Kamasi blue, that molecular pileup becomes highly visible. That dense blue band, along with the distinct pink crystals of the Takayama test, provides the undeniable molecular handshake required to stand up as definitive proof in a courtroom. In sexual assault investigations, identifying the perpetrator's biological material can get complicated if the suspect is naturally sterile or has had a vasectomy. A vasectomy physically cuts the ductus deferens, completely blocking sperm from entering the fluid pathway, but stopping the sperm doesn't stop the fluids. The seminal vesicles and the prostate continue to produce a complex cocktail of secretions loaded with proteins, fluorescent flavin, and high concentrations of acid phosphatase, or AP. To find these fluids, analysts use an AP assay. They introduce a chemical substrate that the acid phosphatase violently breaks apart through hydrolysis. Once broken, the resulting molecules react with an added salt called fast blue B, producing a distinct purple dye. Acid phosphatase is also naturally present in vaginal secretions. To avoid triggering a false positive from the victim's own biology, forensic labs specifically use a substrate called alpha-naphthyl phosphate. This compound is rapidly broken down by the specific type of AP produced in the prostate, but largely ignored by other forms. Finding that prostatic AP is a strong indicator, but the ultimate confirmatory step requires isolating either prostate-specific antigen or semenogelin. To do this, labs use the enzyme-linked immunosorbonphase, commonly known as ELISA. ELISA works by setting a microscopic trap. The lab anchors a base layer of Y-shaped antibodies. When the sample is added, target proteins bind to those antibodies. Then, a second enzyme-linked antibody caps the protein, assembling the complete sandwich and triggering a red signal. But forensic scientists have to measure their samples carefully. If they test a sample larger than 3 microliters, the sheer volume of proteins will flood the system. They bind to all the top and bottom antibodies separately, preventing the sandwich from ever forming and causing a false negative. This is known as the high-dose hook effect. By looking past the absence of sperm and targeting the underlying proteins produced by the prostate and seminal vesicles, forensics can trace the biological reality of sexual contact. a vasectomy defense is rendered entirely useless against the chemistry. The third major body fluid forensic teams hunt for is saliva. Identifying it provides essential context for the DNA profiles pulled from bite marks, discarded drinking glasses, or abandoned face masks. To find it, technicians use the Rapid Stain Identification Test, or RSID. It's a small paper cassette designed to hunt specifically for an enzyme called human salivary amylase. The testing process relies on immunochromatography. The extracted liquid sample wicks up a porous membrane through capillary action. As the fluid travels, it drags any amylase molecules present directly into a trap of dye-tagged antibodies. If the sample contains human saliva, a chemical reaction deposits a distinct pink line in the test zone. Unlike other assays, this specific test won't cross-react with animal saliva, and testing large volumes won't trigger the hook effect. Further up the membrane, a second pink control line will appear. This line proves that the capillary fluid flow worked perfectly from start to finish, validating the test against mechanical failure or tampering. From floors that suddenly glow blue in the dark to microscopic molecular traps hunting for specific enzymes, forensic serology turns basic chemistry into an investigative weapon. It forces invisible biological evidence to permanently reveal its origins, leaving perpetrators with nowhere to hide.